And today we are going to have our PhD student in statistics, Mr. Miles Shi, to present his work on benchmarking doublet detection methods for single-cell RNA-seq data. So to our knowledge, this is the first and most comprehensive benchmarking results we have obtained so far. And before coming to UCLA, Miles had a very interdisciplinary background. He received his bachelor's in computer science and his master's in economics, and then he moved to statistics. So let's welcome Miles to give us this talk.
Thank you for Jessica's introduction. So my name is Miles Xi. So I'm very glad that I can have this opportunity to talk about our recent research topic. And so our topic today is benchmarking computational doublet detection methods for single-cell RNA sequencing data. By the way, I'm a fifth-year PhD student in UCLA Department of Statistics. I'm working with Jessica on single-cell RNA sequencing data analysis. So, so first, let's give a brief introduction about the background. So basically, this is a very brief figure about single-cell RNA sequencing pipeline. So we start from a solid tissue, and then we isolate tissue into single cells, and we try to capture single cells and label those cells, and so that we can identify the RNA in each cell and do the sequencing. So after that, we can obtain our data matrix and do the following downstream data analysis. So the doublet formation actually happened in this step when we try to catch single cell in each, say, reaction volume. But as a result, for some reasons, we catch more than one cell, and we think that's one cell, so we will have issues. So here is a more detailed illustration about how the experiment will form doublet. So basically, there's two pipes, and in these vertical pipes, we have one flow from up to down. This is a cell flow, and we also have a horizontal flow, which is the oil and the breed, the beads. Okay, so when they are intersecting in the middle, we hope that the horizontal flow will form a reaction volume, which includes the bead and the one single cell. And after that, you see we form this reaction volume, and it includes one cell and one bead. And then we insert some labels, say UMI, to this bead, so that we can label each cell inside this reaction volume, so that the following sequencing will give the information on single cell level. However, there are some probability that when these two flow interacts, either flow sometimes will be too fast, so each reaction volume will include two or even more cells. So for the normal single cell, we call them singlets, but for all those reaction volume with more than one cells, we call them doublets. So doublets will have some negative impact on downstream analysis. For example, in cell clustering, all the doublets will form these superior clusters because those cells are very different from normal single cells. And when we do clustering, we will see those cells will be together, but they are not true clusters because those doublets don't exist in the original samples. Or in trajectory inference, we may have doublets in the middle of two branches to form this bridge. So we think, okay, so this is not only a branch, but also there's a branch in the middle, but actually it turns out they are not real branches. So that's all the issues can bias our downstream analysis from doublets. So to deal with this problem, there's two directions. First one is to design some experiments to identify doublets. And the second direction is to use computational methods to find those doublets. So the advantage of computational methods is that we don't have to design special experiment to find doublets, and it will save our time and money. And also, it can help us to identify doublets from existing data sets. Okay. Okay, so currently we have about 10 different computational doublet detection methods. So, and their design are based on different algorithm design and different language, different platform. And in the field, there's no benchmark study that can evaluate all those methods so that we can give downstream users a guidance about which method is better for their analysis. So that's our motivation. We want to do a systematic benchmark to see which method can perform better based on users' needs. Okay, so generally speaking, all the methods can be divided into 5 categories. So the first one is KN-based method, K nearest neighbor based method. So the idea is to find those doublets. People know the doublets are basically 2 cells in 1 reaction volume. So first step is just to simulate those doublets. So from the original data. So people just use randomly choose 2 cells from the data set and either add them together or average those 2 cells to generate artificial doublets. And then they perform dimension reduction to put all the original cells and artificial doublets into a low dimensional space. And then people define a distance so that for each cell, you want to check what's the proportion of artificial doublets within that distance. So the argument is that if that cell is, or we call it the job if that job is a real doublets, then around that real doublets, you should have more artificial doublets, right? And then for the true singulates, you should have small number of artificial doublets. So that proportion will give you the doublet score. So when you have higher doublet score, that cell, that job will be more like doublets. So that's the first method, kind of method. Second category is classification-based method. Still, they first simulate artificial doublets by adding or averaging two cells from your data set, and then they train a binary classifier on your merged data. So they try to classify between the original data set and artificial doublets. And after you train this classifier, you apply this classifier on the whole data set so that you can obtain the probability to be assigned to each category, either a single cell or doublets. And people use that probability as the doublet score. And the third category is statistical test-based method. So basically, people use a different kind of statistic and use the p-value as the doublet score. And next one is deconvolution-based method. So still, you first form artificial doublets, and then you do the deconvolution on singulates, on original data set and the doublets, and try to see based on the deconvolution space, which cell are closer to each other. So if a cell has more similar to doublets, then that's more like true doublets. And lastly, people ensemble different ensemble-based method. People use ensemble-based method to combine 2 methods and average their doublet score, try to improve over the original either those two methods. Um, to benchmark all those methods, we first collected 16 real data sets with experimentally annotated doublets. So this is the most comprehensive doublet data set with doublet annotations, and all those methods are generated by experimental design, so special experimental design. So there are two different experimental protocols here. So first, we benchmark all those methods based on these real data sets. So basically, we calculate the doublet score, and we calculate the AUPRC and the AURC on those real data sets. So those two metrics basically give us the general performance of your binary classification, because in this problem, we essentially do binary classification. We try to differentiate between singlets and doublets. So it turns out that those three methods, solo, doublet detection, and doublet finder, they relatively perform well on those real data sets. And also, we found that from figure B, the performance of methods are very different on different data sets. So some data sets are easier to for method to find doublets, but others are very difficult. Also, because in the reality, people have to define how many doublets they want to find. So here we assume we let different methods find 10, 20, or 40% of doublets from the original data set, and then compare their precision, recall, and the true negative rate. So basically, the conclusion is very similar to this overall detection accuracy. So we realized that there's one method, doublet-detection, did not give you doublet score. It only just give you the predicted labels, which cell are doublets. So to compare this method with all other methods with doublet score, we let doublet-detection to decide the number of doublets you want to identify, and use this number to cut off the doublet score of other methods, so that each method choose the same number of doublets and compare their precision, recall, and true negative rate. So even though we let doublet-detection to decide the number of doublets, it did not outperform other methods. And next, we check how methods perform under different experimental settings and biological conditions. So in this benchmark, we use our in-house designed simulator, IC design, to generate realistic synthetic data sets with with known doublet labels. And we adjust the 4 aspects of our data set: the doublet rates, how many doublets we have, the sequencing depth, the cell type, how many cell types we have, and heterogeneity means how different between two cell types. If you have higher heterogeneity, which means the cell types are very different, otherwise they are more similar. So we found that with higher doublet rates, higher sequencing depth, more cell types, and high degree of heterogeneity, in general, all the methods will perform better. So the y-axis is AURC to measure their overall performance. As you can see here, we have, say, if you have more doublets, then it is better for, it benefits the performance of all the methods. So there's one, I think, so this illustrates that in the reality, people want to catch more cells, then they will have more doublets. But it turns out that the doublet detection method can perform better in this setting. So it encourages people to give higher and higher throughput of those experiments. Then we want to check how methods impact or improve the downstream analysis after they remove doublets. So first one, we want to check the effect of on clustering analysis. Here is a figure about what's the effect, what's the impact of doublets on clustering analysis. All those red points are so basically they are generated from our simulator. All those red cells points are doublets. You see here we have 4 cell clusters, and all those red doublets here, they form superior clusters, and some of them are within clusters. So the goal of those methods, the most important goal is first to remove all those superior clusters so that we can identify these 4 main clusters. And also the second goal is try to remove as many as possible all those within cluster doublets so that we can get pure clusters. So this is the benchmark result. This figure basically we use simulation to generate 4 cell type, 6 cell type, and 8 cell types with each with 20% doublets, and we let all the different methods to remove 1%, 2%, up to 25% cells or doublets. Then we use Leuven clustering to identify how many clusters we have after we remove different number of clusters. So so the yellow grade means the Leuven cluster correctly identify the correct number of the 4 clusters or 6 or 8 clusters. Otherwise, if it is wrong, you have orange here. So basically, if you have more yellow grades, which means you have better performance. So in this benchmark, we found that doublet finder and doublet detection give us most correct number of clusterings. And this figure shows among those correctly identified clusters, what's the proportion of singlets, which means among all those gray clusters, you want the gray ones, the correct singlets, as many as possible. So still, these 2 methods perform better, but we also found screwblades perform well in this study, even if it did not give us most correct result in the number of clusters, but it actually can remove many of those doublets from the true clusters. And then we benchmark the effect on high identification of highly variable genes. So basically, we first identify highly variable genes using a Surette on clean data, and also identify them on contaminated data with 10, 20, or 40% doublet rates. And we use each method to remove the correct number of doublets and use the Jacob index to measure what's the overlap of identified highly variable genes between after removing data sets and the clean data set. So if you have higher similarity, you should have higher Jacob index. So we found that only doublet finder the screwblade outperform with the negative control, this contaminated data. So only these 2 methods improve the identification of highly variable genes. So next one, we check the effect on DE gene analysis. So basically, still we use simulations with true DE labels and use different methods to remove certain number of doublets and check the precision, recall, and the true negative rate of DE genes using 3 commonly used DE analysis method. So it turns out that the impact of the negative impact of doublets on DE analysis is not that significant as before, as previous analysis, and the main effect is on recall. And still, doublet finder and doublet detection has the better performance in this benchmark. And the last downstream analysis we did is the trajectory inference. We simulate two scenarios. First one is we have two branches here, and then we have sequential branches with three branches. So, and we simulate certain number of doublets you can see here on the original data set, original contaminated data set. The red doublets bias the trajectory inference, right? So you see here we have a superior branch in the middle, and all the methods, most of the methods can remove these superior branches by removing all those red points, even though the degree of removing is different, but most of the methods can remove this branch. And also, most methods can remove the superior branch in this sequential case. So only one method failed to remove the branch. And then we also conduct a temporarily differentiate gene analysis. So, and find that basically methods have similar performance except the doublet cells. So this method didn't perform very well in this benchmark. And next, we try to compare the computational efficiency of different methods. So first one, we check the distributed computing. The idea is that if we have a very huge data set, say millions of cells, it's very difficult to run all of them on one computer. So in that case, people want to use distributed computing to divide them into small batches and use different computers to calculate all those small batches and then get different scores, doublet scores, and combine them together and do the following analysis. So we simulate that scenario by divide the data set into 2 up to 10 batches and calculate and merge them together, calculate their performance. We found that when we divide into more batches, the performance actually is going down, which means in each small batches, all the methods lose some global information. But there's one exception, which is CXDS, which is almost flat on this scenario. So this green one. So only this method can be resistant to this information loss. And then we can compare the speed, scalability, and the stability. So basically, the conclusion is that the best method, doublet finder, is relatively slow, and the fastest method, CXDS, is not the best in terms of accuracy. So there's still some room to improve into this direction. And then we also summarize how user-friendly of those methods, and we create a score system to summarize. So basically, still doublet finder give us most user-friendly documentation and implementation. So lastly, we want to summarize and give some suggestions. So basically, based on our benchmark, we found that doublet finder has the best detection accuracy, and CXDS has the best computational efficiency. We found that there's still some room for methods to improve, because we found that the best method, doublet finder, has only average AUPOC 0.537 on all real data sets. And also we found different protocols actually have a different method, have very different performance on different protocols. So we summarize all the performance on these 4 different experimental designs. So those designs gave us true doublet labels. And there are some open questions that the field may want to explore further. First one is how to accurately estimate the unknown doublet rate in a data set. So because most methods can give us doublet score, but for users, you have to decide how many doublets you want to find and get a threshold on those doublet scores. And if you have doublet score higher than that threshold, you call those cells. But how to accurately define find this number? So there's some heuristic explorations for different methods, but there's no guarantee you can find correct numbers. And then second one, how to distinguish between homotypic doublets from singlets. So homotypic means the doublets are formed from two cells from very similar say cell types or clusters. So in that case, the doublets is very similar to singlets. So it's not easy for the method to find them. The next question is how to better generate artificial doublets, because almost every method use artificial doublets to help them identify true doublets. So basically, they just add up two randomly selected cells. So the problem is, on average, you will double the library size and the number of expressed genes for those artificial doublets. But we actually check the real data set and find that the true doublets usually have 1.5 times of library size and the expressed genes. So which means new methods can simulate more realistic doublets based on this ratio. And next one, we check the relationship between different methods. We calculate the Pearson correlation between two methods doublet scores, and also the Jaeger index of those identified doublets. And we found that actually the top performance methods are actually more similar in terms of those two metrics. And then lastly, we check the effect of hyperparameter tuning on the performance. So we found Scrublet and doublet funders are very similar in terms of their design. They both use KN, K nearest neighborhood method, but their performance are very different. So we found that the difference partly because they use different hyperparameter tuning to train their classifier. And so we substitute the Scrublet's hyperparameter by doublet funders hyperparameter and check Scrublet's performance. We found that in some data sets, Scrublet gets some improvement, but not on all data sets. So we suspect that the hyperparameter tuning has some effect on the performance, but not all of them are due to the tuning. There are some other implemental details. Lastly, I want to, so also this work has been accessed by Self Systems, and I also want to thank Dr. Jessica Lee and all the members in the JSB groups. So, and I would like to take questions and comments. Thank you.